chemical or some hormone, and they will get trapped into this particular trap. So they are funnel shaped. That's why they're called as funnel trap. So you gather them here, and after some say some 15 days or 10 days, you can just take this out, go to the lab, and you can uh, look for what kind of animals are present in your sample. Okay, now next is called as hand capture, right? So in hand capture, what you do is you actually go and you catch hold of the organism. Like now, this is commonly done for frogs, commonly done for reptiles, say snakes and lizards. So the herpetofauna specialist, they go to a water body or they go to a grassland or rocks, and they go and they catch hold of these animals, especially at night, okay, or immediately after rainfall when frogs will gather to a particular area, and maybe they will attract them to a particular area and then they'll go and catch them, or they'll go to the animal itself and they will catch them. Maybe they will put some light, or maybe they will make some sound, okay, or maybe they will imitate a call of the opposite sex, and they will go and catch hold of the animal. So this is called as the hand capture technique, mostly used for amphibians and reptiles. Okay, now after you catch these animals, the thing to remember is, if you are counting it, if you're looking for the population, or if you are looking at the abundance, you should not repeat the specimen that you have collected. That's why you go for a technique which is called as marking technique. Okay, so you capture an individual and you mark it, okay, or you mark it visually so that you can monitor that particular organism, where it is moving, or which area it is going. Right. So these techniques, called as wildlife marking techniques, are used to mark and identify individual animals in the wild, which helps us to study the behavior of that particular animal, which helps us to study uh, the population changes, and which also helps us to study their relationships, or what we call as ecology. Now, these marking techniques can be visual, where you if you must have if you've been to villages, you have the cow, the goats, then the buffaloes, where they will mark that particular individual, so that this individual, based on the number or based on the barcode, we know the information about this individual. So you can do it for wild animals as well. So this is called as ear tagging. So you tag the ear with a number or with a colored tag, or you can have a collar with an identification tag, okay, with a GPS device helping it to track its movements, or even a radio transmitter around the animal's neck. So what we can do is we can understand their behavior, where it is going, when it is feeding, when it is sleeping, when it is hunting. All the behavior of the animal can be studied by marking this animal and following the animal by sitting in a lab. Sometimes you even bandage the animal using a colored bandage or a numbered bandage, which you can put around the leg of an animal, or maybe if the tail is long, you can put it near the tail of the animal. Or if you look at the sheep, you can apply paint, which doesn't go for say some 30 days or 40 days, permanent dyes to an animal's fur, feather, or even scale. So for birds, you can use this technique, or for mammals also you can use the paint marking technique. Now, why do you do it? To monitor the diversity, to monitor the population. Okay, now these are visual marking techniques. We also right now have something called as electronic marking technique. So there was a movie called as Azoba long time back, which came in 2014, where the movie was about a leopard which was moving around in the Junnar area. So they were tracking it electronically using radio telemetry, where you attach a radio transmitter to an animal, allowing us, you PhD students, researchers like us, to track the movement of that particular animal. Now, this is mostly used for larger animals, mostly predators, and you can also fit a GPS tracker to an animal, enabling us to monitor its movement and also what kind of habitat it is using. Say for leopards, nowadays leopards are also using sugar cane, right? So if a particular individual is using sugar cane, it is more prone to be found near human habitation. So based on where the animal is spending a lot of time, based on radio telemetry and GPS tracking, we can even understand the behavior, and we can avoid unnecessary confrontation with these predators, which may turn aggressive during some times. Okay, so these are the different ways in which you can look at terrestrial habitats. Okay, now we move to the next point, that is aquatic habitat, which is my expertise. So I work on aquatic habitats. I work on permanent habitats, temporary habitats, small habitats. I also work on huge habitats. So if I'm working on invertebrates of habitats, these kinds of habitats, we have to monitor the diversity and abundance of these organisms in the water body. Okay. Now the problem is we cannot enter inside the water body, right? So the thing is, you can enter inside a forest, you can walk on the road, you can walk along a designated path, but it becomes extremely problematic for studying the zooplankton, or for studying aquatic insects, or for studying the fishes. Right? For that, you have to go to the water body, or you have to use some advanced equipment called as sampling gear, which will help us to have a look at the diversity that is present within the habitats. Now, when I speak within the habitat, it not only means at the surface, it not only means it not only means near the surface, or it not only means near the shore, right? It also means from the center, it also means from the bottom, right? So if you are looking at benthic organisms, right, looking at surface organisms, right, you have to use different kind of equipment to do the sampling. Okay, right. So we need to sample at various depths, right, to understand the diversity present at the surface, to understand the diversity present at the shore, to understand the diversity that is present at the bottom, right? So there are many different kinds of techniques that we use to sample fish, amphibians, as well as aquatic invertebrates, and even zooplankton. Okay, now this is a slide where you get to understand the different generalized sampling technique methods that you can use for marine organisms. Okay, now so this is the sampling device or the method, and these are the targeted organisms. So the most commonly used technique or method is the use of nets. Okay, so here what you do, you use nets.
बघ कसं पकडूनच चालली आहे.
Second. Okay, so here we use nets of different mesh size, and these different mesh size can catch different kinds of organisms. Say if you use a net of mesh size means whole size of, say, some say higher 50 micron or 10 micron or 20 micron, you can go for catching phytoplankton. If you use larger size, you can go for catching larger nectons. Okay, so you can use a dip net, or you can use a gill net. You can even use traps. Okay, we'll talk about them in the further slides, to catch fishes and crabs. You can also do something called as visual observation, especially of animals which you cannot catch hold of, like say jellyfishes. So you can just monitor the bloom of jellyfishes, or you can just observe the shoal of fish, or you can use some cameras, underwater cameras. Okay, or you can use scuba diving methods to understand what kind of animals are present, especially the sessile organisms, that are the corals that are present inside the water body. Okay, so you can use cameras, you can use video cameras to record and understand what kind of animals are present in water. Okay, you can also do aerial survey, where you can go up in a helicopter, or you can have a drone which can help you to understand organisms like whales or sharks, which are easily visible from above. You can also go for acoustic or echo sound method, where you can look at marine mammals or some big fishes. For benthic organisms, you can use dredges or sledges, which can be put on in the bottom, and you can pull it towards yourself, or you can pull it above, and you can get the organisms that are present in the soil or in the sediment, right? So you can even use remotely operated vehicles, where you send them at the bottom of the sea or bottom of the ocean, or maybe of the reservoir, deep down inside, and you can look for organisms present inside. Light traps, very commonly used for photosensitive organisms like plankton or organism that can move. So you can place this inside here, and you can switch on the light, and they are attracted to the light, and you can collect these organisms, or you can just photograph these organisms uh, without even going inside the water body, right? So you can even tag and mark bigger organisms like whales and mammals, and even large benthic fauna like the corals, right? So now we move to the different ways in which you can collect aquatic organisms. Okay, so this is the first way. So for plankton, plankton means organisms which are microscopic, organisms which do not swim on their own, organisms with microscopic sizes which you cannot see with your eyes. So you can't use visual method. So what you have to do is you have to catch hold of them from the water body that they are present inside. So what do we do? We use a net made up of nylon or made up of silk or made up of cotton. Most commonly we use nylon because it doesn't shrink and the whole size doesn't become smaller. Okay, so what we do here, we put the net, you tie a thread to it, you have a container here, and you put the net inside the water body, you put it down, and you pull it up, or you just throw it and you pull it to yourself, right? So as the net is pulled towards you, water will enter inside the net, and water will come out through the holes that are present on the net. Okay, so water, even if the pore size is very small, water will come out through the net, and all the organisms that are bigger than the pore size of this net will get collected inside the bottle that is here. Okay, so you can do diversity studies, okay, where you don't have to actually count, uh, where you actually don't have to count the number of individuals. You just have to understand the diversity, or you can measure a fixed amount of water and put it inside this net, and then collect your sample, and that can help you to understand the abundance, diversity, as well as abundance of the plankton that you are studying. So you can use it for phytoplankton also, you can use it for zooplankton also. So based on the mesh size, you can choose your organism of study. So for example, if I'm looking at rotifers, I need a smaller mesh size net, which will help me to collect rotifers which are very very small. Okay, so they are around 80 micron and 100 micron. So anything bigger than 53 micron will be collected in the net and will get collected into the plankton bottle, which then you can preserve in the field, or you can bring it back to your laboratory and observe directly under microscope. For bigger organisms like cladocerians, you can use bigger mesh sized net. For phytoplankton, you can go for 10 micron or 20 micron, depending on the group of your choice. Okay, so the mesh has holes that are large enough to allow water to come out, but small enough so that the plankton remains inside the net, and that plankton you can go to the lab and study under a microscope. Okay, now sometimes you can see the organisms, right? Especially in shallow water bodies like ponds or streams, where you can see insects, where you can see molluscans, okay, or other animals. You can just walk. Here you cannot walk in the water body because it is deep. You have to be on the shore, or you have to take a boat. Here you can walk in the water body, and as you see the organism, you can collect the insect, or you can collect the organism. This you can use even for aquatic plants or hydrophytes as well. So these nets can be of various size, various shapes, depending on what is your organism of choice. Okay, so and the length of this can also change depending on where you want to sample. If you're interested only along the shores, you can have small length. If you are interested in the center also, you need to have long length of this particular rod, okay, which can be telescopic or which can be fixed length as well. So we call them as dip nets. Okay, they can be of big mesh size, right? So these are of small mesh size. These can be of bigger mesh size. Okay. Now, right. Now moving forward. Right. So these are called as traps. Okay. So what do? You leave these traps along with some food or some attracting chemical into the water body, and the organism of your choice, say crabs, will enter inside it. Once they enter inside, they will not be able to come out. Likewise, you can use these kinds of net traps, where a fish will enter inside, but it may not be able to come out again. Okay, so you can leave it in a particular place in a water body. You can leave it for a day, or you can leave it for some few number of days or a week or so, and you can come back after some few days and you can check the organism that have been caught in that particular net. Okay, so these are called as traps. So you trap the particular animal inside so that it is not able to leave. Okay, now sometimes what happens is, right, so this net, right, so if you're only interested in diversity studies, this net will help you if you go to a water body, right? And if it is difficult for you to carry a fixed volume container. So as I said, if you have 20 liters of container, you can put 20 liters of water inside, or say 1 liter of water inside, and you can also get the abundance, right? But only at the shores, only at the surface, right? But if you are interested in sampling from a particular depth, say you want to understand the diversity of organisms at the surface, you want to understand the diversity at 5 meters depth, you want to understand the diversity at 10 meters, and so on and so forth. Okay, and as you go down, okay, as you go
वो
down,
मतलब पिक्चर में तो ये दिखा है इतना बड़ा ये वाला है
right, so as you go down, you will not be able to use a bucket, or you will not be able to use a fixed volume container, okay, because that is not open inside. So we have a sampler which is called as Van Dorn sampler. Okay, so what is this? This is a water bottle. This is just a water bottle of fixed volume, and which has opening on both sides, or one side, and it can be used to send this container down. So there is a rope, and there is something called as a messenger, right? So what does this messenger do? When it goes down, it attaches itself and it opens the doors of this bottle. Now, as the doors open, water will enter inside. You pull the messenger, the lids will close. So I can sample for zooplankton or phytoplankton at the surface. I can sample at 5 meters exact. I can sample at 10 meters, and I can get a vertical sampling. Okay. So normally using a plankton net, you can do something called as horizontal sampling. Using samplers, you can do vertical sampling. Okay, so with a capacity of say 1 liter or 2 liter, okay, you can go for microbiological sampling, you can go for vertical or stratification sampling. You can also go for heavy metal analysis along with the diversity of organisms that are present in the aquatic habitats. Okay, so this is a more sophisticated instrument which we normally don't use in freshwater techniques. Mostly it is used in marine water habitats. Okay. Now we have a sophisticated method of understanding diversity, and this method is called as remote sensing. Now the word itself tells you remote means it's far away. Okay, you don't go to that place. Now here you go to the water body to collect the specimens, and so is the case here. So is the case here. So all these types of sampling, you have to be in contact with the biodiversity. But in remote sensing, you can sit in a given area very far away from your ecosystem or from your habitat, and using satellite imagery or using satellite photographs or aerial photographs, drones, simply what we say, you can use this method to assess biodiversity over a large geographical areas. Okay, so if you remember quadrat sampling, if you remember transect sampling, line transect sampling, you have to walk say 100 meters or 1000 meters, and that is time consuming. It is very difficult to go through the forest area, but remote sensing will overcome all these difficulties, and you don't even have to go to the habitat to understand what kind of plants are present in the given area, what kind of trees are present in the given area, what is the age of the trees and plants in the given area, what is the structure of the habitat, what are the changes in vegetation that have happened over a period of time. Say the plants will change over the seasons. So in winter season you will have different kinds of plants, in some summer season you will have different kinds of plants, in rainy season you will have totally different kinds of plants. You don't need to go there to understand what kind of plants have come there. You can just sit in your laboratory or in your office and you can understand it over a large geographical area. So what happens here? There is a satellite, right? And you have the energy source, and this energy is reflected by each and everything that is present on the face of the earth, including buildings and roads and soil. And each and every individual object or animal or plant or organism gives different kinds of energy inputs, which are detected by the satellite. Okay, and based on that you can understand if this is grass, or if this is a tree, or if this is a particular type of tree depending on the color of the leaves. Okay, if this is a water body, if this is a freshwater body, if this is a marine water body. And via the processing station you can analyze, right, what kind of forest cover, you can analyze land cover, you can analyze the changes in vegetation over a large period of time. Remote sensing has been extensively used in botanical studies, used by forest officials to even understand uh, what do you say, wood cutting by poachers, right? So it can also be used for something called as habitat mapping. Okay, so to understand the different types of vegetation and to understand the land cover. Okay, so what is habitat mapping? It's a process of identifying distribution of habitats in a specific area. Now these habitat maps can be used to identify and manage critical habitats for various species. Okay, so based on the number of species present in that habitat, based on what kind of plants are present in that habitats, you can make a priority area for conservation. Okay, so if the abundance is there, if the diversity is good, you can leave that area for the time being for conservation activity, so that you can concentrate more on vulnerable habitats and sensitive habitat. This can be easily done by using remote sensing. So if you look at this particular map of a forest, so this is the inset map. So you can see this is the buffer zone. This is the railway corridor which is passing through the area. This is, and this is the wildlife protected area, right? You can understand what is the forest area, what is the buffer area. You can understand, is it shrinking, is it increasing, just by looking at it from above, sitting in a laboratory. So that is the benefit of remote sensing, right? So you can use it for habitat monitoring along with habitat mapping. You can use it also for habitat monitoring. What is happening to this buffer zone? What is happening to the forest area? Is there any activity in this particular area? Are the trees being cut here? Are the animals moving from herd of elephants moving from this place, or is it moving from this place? Are they traveling out of the forest? Are they entering inside human habitation? Everything can be understood by remote sensing. You can even go for species distribution modeling. And based on SDM, we can predict distribution of species based on environmental variables. Okay. Next is very important thing that is becoming a very subject of many studies is called as invasive species monitoring, especially plant species like lantana. They are increasing in aquatic habitats. Fishes, exotic species are increasing in number. This can be easily monitored from above, especially plant species can be easily monitored, and we can understand, is it becoming a threat to the biodiversity. So these changes in vegetation patterns can be monitored using remote sensing, and you can identify areas where invasive species are becoming established, they are increasing in number, and then you can develop strategies to control these invasive species and even prevent their spread. So you don't have to go to the particular ecosystem to understand, is it increasing in number or is it decreasing in number. Now this is also a very important application of remote sensing, where you can understand the forest fire incidences, you can understand the spread of forest fires, you can understand if it is getting extinguished at a particular area, where are the spots of the fire, and how to control and from where to control the forest fires. Forest fires are becoming a huge menace to the biodiversity, especially in areas, forest areas which are near to tourist destinations or villages, where people go into the forest areas and inadvertently or deliberately cause forest fires, which may result in drastic reduction of the biodiversity. So if the grass is all burnt away, if the trees and if the herbs and shrubs are all burnt away, the wildlife may also suffer because of forest fire. Remote sensing is becoming a great boon in understanding forest fires occurrences and also in controlling the forest fire. It is also helping us to understand and monitor wetlands, coral reefs in marine habitats, detection of wildlife poaching, especially with respect to animals like tiger, lion, and elephants, assessment of land use change. So if there is a forest, and is there agricultural practices intrusion happening near the forest, is there some tourism activity, illegal tourism activity happening inside the forest, this can also be understood by remote sensing, and all this helps us to understand biodiversity and protect biodiversity. We can also monitor water bodies, especially reservoirs and dams. We can have a look at their volume. We can understand if the dam is about to break, or any illegal activities happening near the reservoirs. We can, and as I told you, the most important thing, we can detect forest fires and we can control or manage forest fires because of remote sensing. And finally, we move to the latest innovation in biodiversity analysis or assessment or monitoring, that is called as genetic analysis. Very commonly we call it as DNA barcoding or DNA sequencing, where we sequence a short fragment of DNA, even from a hair, even from a nail, or even from a microscopic animal. We can get a short fragment of DNA, we can amplify it, and based on the barcode that we get or based on the sequence that we get, we can identify the species, even without looking at the individual species. Okay, so just from any part of the body of the living organism, say for plants or microorganisms or invertebrates, or even for bigger animals, even from their scat, even from their hair, okay, even from their urine, you can get a DNA and you can amplify it and you can identify animals, right? Also, you can identify animals which are very difficult to be identified based on morphology alone. Okay, so in the end, I'll show you one example that we used, where we use genetic analysis to separate a species which was very difficult to be identified morphologically, right? So DNA barcoding, genetic analysis is becoming a great boon in biodiversity analysis and also for conservation. So what do you do here? We create something called as barcodes, which are unique identifiers, like what we get in the supermarket. So if you go to DMart or if you go to a Reliance store, they have a barcode for each and every uh, instrument or equipment or grocery item that is placed in their center. You just have to scan it, and based on the barcode, you can get the information of what it is, what is the weight, what is the price. Okay, so we create unique barcodes for animals. And this method is very fast, very quick, very cost effective. The thing is, you just have to get a single specimen and you can tell the difference between two species very easily based on this unique method called as DNA barcoding. It can also help you to identify